Dynamic filaments of proteins, called microtubules, have several important roles inside cells. They provide structural support for the cell, they help to pull chromosomes apart during cell division, and they guide the trafficking of proteins and molecules across the cell. The building blocks of microtubules, called alpha and beta tubulin, are continually added to and removed from the ends of individual microtubules, causing it to grow and shrink. Microtubules display multiple modifications, such as deuterocination, the removal of the C-terminal tyrosine from alpha tubulin. Deuterocination of tubulin is crucial to microtubule dynamics and functions, and its defect has been implicated in cancer, brain disorganization, and cardiomyopathies. Although the deuterocination reaction was first described 40 years ago, the identification of the tubulin tyrosine carboxypeptidase in charge of deuterocination remained unsuccessful. To isolate tyrosine carboxypeptidase, we developed a thorough purification process to enrich this elusive enzyme lowly expressed in tissues. To select the right protein, we collaborated with chemists from Stanford University who developed a small molecule that was used as an irreversible bait. Thanks to this innovative approach, we identified Vasohibins as enzymes that perform the tyrosine carboxypeptidase function and found that their small interacting partner, called small Vasohibin binding protein, is essential for their activity. Indeed, we demonstrated that Vasohibins, when complexed with small Vasohibin binding protein, exhibit robust and specific tyrosine carboxypeptidase activity on microtubules. To confirm the functional significance of Vasohibins and their role in alpha-tubulin deuterocination, we assessed the effects of reduced production of these proteins in differentiating neurons. Our results showed that reducing the production of Vasohibins or small Vasohibin binding protein in mouse cultured neurons reduced deuterocinated alpha-tubulin levels and caused severe differentiation defects. We next tested the functional significance of Vasohibins in vivo in the mouse brain and showed that the reduction of their expression disrupted neuronal migration in the developing neocortex. Overall, our results indicate a critical role of the Vasohibins and their associated partner small Vasohibin binding protein in neurons and the brain, and further support that they represent long-sought tubulin carboxypeptidases enzymes. The impact of deuterocination enzymes on microtubule functions in health and disease may now be further explored.